afternoon. In 1880, in 1880, TB bacteria, which is Mycobacterium tuberculosis, was described. Yeah. Since then, mortality and morbidity numbers are very high. In 2020 only, an estimated number of 1.5 million TB patients died, and another 10 million active TB patients infections added the same year. The map on the right there shows the distribution of TB cases around the world. And it's clearly that countries in Asia and Africa accounting for the highest numbers of TB infections. But to be more specific, in Europe, the same year of 2020, there were around 164,000 TB cases. And it's worth saying that these numbers are for active TB cases. So patients are contagious and they have symptoms. If I want to speak about latent stage, where the bacteria will be isolated within nodules, as seen in the panel on the left, so the numbers will be a lot higher. To be more specific, in the UK, I'm trying to move to the next slide. Okay, thank you, John. Last year, there were confirmed cases of 4,500 TB, active TB cases. The panel on the left shows the distribution of cases around the country. And London accounts for 35% of the cases, with a very high rate as well in the West Midlands. It's very interesting if we look at the right panel in the first quarter of 2022, the number of TB cases are very comparable to 2021 and to 2020, which means that we need efficient drugs just to solve this problem. So currently, TB is treated by initial phase of 4 drugs to be given orally for a period of 2 months, then a continuous phase of 2 drugs orally as well for another 4 months. The challenge, actually there are 2 challenges for this. The first one is for these drugs to be given orally, because these drugs have a very poor bioavailability, and they have a very short elimination rate, which means that these drugs will be eliminated very quickly from the body, and we need a lot more doses to be given in order for these drugs to be going to the site of infection. The table next to me here shows the side effects of many of the first-line TB drugs, and we see if we increase the doses, the chances of having liver and kidney toxicity are higher. The other challenge we have within this treatment, that giving the drugs for 6 months is very hard, because what happens is patients during the initial phase, they tend to feel better and they stop the treatment. And that's why, and because it's an antibiotic, when you stop the treatment before taking the full course, the bacteria will not become susceptible to the drug anymore, and they will become resistant. And that's why in 2020, there were 500,000 patients developed multi-drug resistance or rifampicin resistance. And the other option will be is giving another drug for 12 months. So the aim of my project is to overcome this problem by designing a direct inhalation therapy going to the site of infection in the lungs. And this therapy will contain many first-line TB drugs, and also an antibacterial agent, which silver in my case. And this is just to help killing the bacteria. The third aim of my project is to design a model containing type 1 alveolar epithelial cells, type 2 macrophages, and also the bacteria itself, just to make sure that our system is safe, but also efficient. So to start with, I started making the silver nanoparticles. I started from the charged silver, and using reducing agent of sodium borohydrate, I tend to neutralize the silver, which will start to aggregate together. And then using a carrying agent of sodium citrate, tend to have this citrate charges around the silver to protect them. Then I did several characterization of morphology, size, and compositions. The panel on the left for TEM shows a tiny spherical sizes of particles, which are purely silver, thanks to the EDX experiment, and with a mean size between 15 to 18 nanometer. The zeta charge, which is the surface charge, so showed minus 25, which means a very stable nanoparticles. Then I did a cytotoxicity using two different assays, which is MTT to assess the viability of the cells, assessing the mitochondrial activity, and also LDH to see the effect of the particles on the cells, whether there is any necrosis or no. And I used three different types of lung cells. So I used a transformed type 1 cell line and a primary type 2 and macrophages. Cells were exposed to silver for 24 hours and with different concentration of silver. So if we look at the first result, so the NTT, which is the viability, is on the left. It shows that up to the concentration of 10 microgram ml, silver didn't show any toxicity on these cells. Higher concentrations of 25 and 50 showed toxicity. And the same results are very comparable results with the LDH, and instead that the y-axis is different. So there it's a viability, and the other one is exactly the opposite of cytotoxicity. And it's very interesting if we look into panel B, we look how these tiny dots of silver were taken by the cells. The same with type 2 alveolar cell line, primary cells. Also, we saw that concentration up to 25 microgram ml was safe. Again, with macrophages, the same 10 microgram ml was also safe. And again, the silver was taken by the macrophages. This experiment was very important in order to know the treatment window of our silver nanoparticles, and that was added. And before adding the silver, I did another experiment to see whether silver is able to have any kind of a pro-inflammatory response when added to the cells. So I did measure the IL-6 and IL-8 cytokine using ELISA, and I see as well for all the concentration less than 25, the levels were very normal to the control, which now I'm sure that any concentration less than 10 microgram ml should be okay. And that's why I started to combine silver nanoparticles with rifampicin using a PLGA polymer. The method I used, it's a solvent evaporation technique. And the reason I used it because the drug is water insoluble and I want just to formulate it together in order to have oil and water emulsion. And then by evaporating the organic phase, I get the PLGA microparticles. The same characterization happened for this system as well. So the SEM image showed that all of the particles, they have a size of less than 5 micrometer, which means they are in the respirable size. I found also a very high encapsulation rate of rifampicin. The rate of silver was good as well, of having 53.62. The next step will be now what I'm currently doing is measuring the cytotoxicity of this system using the same cells. And also at later stage, I'll be using TB bacteria alone and infected cells with TB bacteria to see the efficacy of this system. Then the step later will be by doing an aerosolization of this system to make sure that I'm able to deliver it to the site of infection. Because I got really good results, that made me also trying to include another drug to the system, and I was successfully able to have many good results on that. Hopefully next time I'll be able to speak more about it. Finally, I would like to thank my both supervisors, Terry and Alex, and all the people who helped me throughout this project. Thank you.
Do you have any questions online or in the room? I should suggest that once you come to a name, very important. Hi, great talk. Thanks, Darryl Hill, University of Bristol.
did you see uniform uptake of the particles by the macrophages, or did you see a subpopulation that were taking that more preferentially? Because it looked like some cells were really loaded.
Thank you for your question. I think it was a little bit related to the concentration of the silver applied to the macrophages, because at higher concentration of silver, it was a bit toxic. So yeah, it was hard to see if it was uniform. At a concentration of less than 10 and 5, yeah, the distribution of particles taken by macrophages were uniform. However, this could be done in a better way by having kind of SEM images of different macrophages taken there to make sure like the amount of silver going inside the macrophages. I hope this answers your question.
Sharon from University of Cambridge. That was a nice talk. I was just wondering, what about post-antibiotic effect? Are you looking to see whether these silver nanoparticles are retained in the main itself for significantly longer periods of time than kept in the room?
So, are you asking about whether I would have a stability study of silver, or are you asking about the antibiotic? Or what?
Well, both. So it's 24 months in barrels.
So
after uptake, how long are these particles being retained?
So what I will do different timelines. So I will have like ones to be done after, for example, 24 hours, then another one after 72, 48 hours, to make sure even at this concentration, they still take a longer time. So yeah, this is something I'm working on.
Thanks very much, Khaled.